our slot is TR, no?
Yes, ma'am.
Class
Yes,
slot
ma'am.
TR. So according to that new announcement, the revised exam schedule, TR class, the last class is 14th. 14th December is the last class. And then the final exam is on 19th.
Yes, miss.
Ma'am,
So
22
your
no?
final.
Ma'am, 22.
Oh, sorry. Yes.
22.
I'm sorry. Yes. Your class slots are SR. Yes. Your class
Oh,
slots
ma'am.
are
No,
SR.
No.
TR.
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 TR, ma'am.
Ma'am, TR2.
TR. Yes. If it is TR, then the last day of class is 14th December. And then the exam date is on 19th.
I'm 22.
So no, TR is 19th.
G miss 9.
টিউসডে হরতাল থাকে না তো?
Yes, that's why they put it on Tuesday. Okay, so if it is on 19th, actually that means we have only 2 weeks, no?
Yes, ma'am. Only 4টা class baki.
We have only 2 weeks after this class. So we hope that in 2 weeks we should be able to complete 4 lectures.
4 লেকচার কমপ্লিট করে ফেললে মিস আমাদের পরীক্ষার আগে এতগুলো লেকচার মিস।
Like it is mentioned in the notice that if the faculties they have to complete or they must try to complete the syllabus, and if they need to take extra classes, they should take extra classes. And here I am not taking any extra classes, but I must target, like after today's class, at least 4 more lectures I should complete. Okay, 4 more lectures at least. We should complete our metabolism part. And the very last lecture is about the plasma membrane and the transport system. We can skip it because I think in 105 you actually learned about plasma membrane and the transport system. But I think I must complete the anabolic phase. Okay, otherwise this course is meaningless.
ম্যাম আমাদের তো
But
ক্লাসরুমে
now the
অ্যাসাইনমেন্টের
question is, sorry,
ম্যাম আমাদের তো অ্যাসাইনমেন্টের ডেডলাইন দেওয়া ছিল 21 এ, সেটা কি আগায় আসবে নাকি সেটা 21ই থাকবে?
okay, now the final grade submission is 30th. Let me check again.
মিস তাহলে কি আমাদের প্রেজেন্টেশনটা হবে?
30th, let me check. Yes. Where is that grade submission? Yes, 30th December is the grade submission date. So in that case, I think what was your assignment submission date?
21 ম্যাম। 21 মিস।
Then I will keep it 21, or maybe because according to the notice, our exam is on TR 19th December. So if it is 19th December, you can submit it 21st, or even I think 23rd is still okay. Assignment submission.
Okay, miss.
Okay, so let me revise it in the assignment work as well, so it will be easy for us.
ম্যাম আমাদের মিস প্রেজেন্টেশনটা তাহলে
Where is the assignment? Let me come. So your assignment uh 23rd, okay, 23rd December. Okay. Okay, now about the presentation. And so how many courses do you have? I don't know, maybe some of you have more than 3 courses.
Yes, ma'am.
Hmm. So like if I target that we will have the presentation on 23rd,
মিস আমাদের
23rd
এক্সাম
or
আছে তো মিস ওই দিন ফাইনাল এক্সাম তো আছে।
থাকে ওই দিন।
আমাদের এক্সাম আছে ম্যাম।
ইয়েস ম্যাম 23rd একটা এক্সাম আছে।
okay, so then maybe 26th Tuesday.
মিস আমাদের প্রেজেন্টেশনটা বাদ দেওয়া যায় না মিস, কারণ আমাদের উপর অনেক প্রেসার পড়ে যাবে মিস। একসাথে অ্যাসাইনমেন্ট, প্রেজেন্টেশন, দেন আবার এগুলো সিলেবাস
জি
শেষ
মিস,
করা।
সিলেবাসটা তো শেষ করে এক্সামটা
Yes,
দিতে হবে।
Because if I don't do the presentation, how I am going to give you the mark? Then I have to take the final exam maybe out of 30. Do you agree on that?
নো ম্যাম।
ওকে ম্যাম।
Yes.
মিস সমস্যা
ম্যাম
নাই।
এই সিচুয়েশনে করা, হ্যাঁ প্রেজেন্টেশন থেকে ফাইনাল এক্সামটা বেশি মার্কস দেওয়াই বেটার। জি মিস।
ইয়েস মিস, কারণ আমাদের এই সিলেবাসও কমপ্লিট করতে হবে মিস। ফাইনালটা আমরা
ম্যাম
বেশি মার্কস
অ্যাসাইনমেন্টের মার্কসটা যদি 5 মার্ক ইনক্লুড করা হয়।
No, no, no. I am not just going to put more marks in assignment. I will do better, add it to the final exam. Okay.
ম্যাম তাহলে প্রেজেন্টেশনটা আমরা অনলাইনে দেই।
It's not about online or whatever it is. So I am just proposing you, what about 26th?
ম্যাম 26 ঠিক আছে,
26th,
কারণ সবারই তো 23 তারিখে এক্সাম শেষ। 3 দিন তো এক্সট্রা পাওয়া
yes,
যাবে।
26th is a Tuesday and I have to submit your grade on 30th. So I have time to assess you and submit your grade.
ম্যাম তাহলে ফাইনালের মার্কসটা বাড়িয়ে দিলেই ভালো হয়।
মিস ফাইনালের মার্কসটা বাড়িয়ে দিলেই ভালো হয় মিস।
না ম্যাম 26 তারিখ প্রেজেন্টেশনটাই ভালো হয়।
ম্যাম প্রেজেন্টেশনটাই ঠিক আছে 26 তারিখ, কারণ 23 তারিখে তো এক্সাম
You
শেষ
people
ফাইনাল।
decide. You people decide on that, because I am not just going to give increase the mark of assignment. I don't agree with that. Okay, so 26th, I think it is a good date. It is a good date. After you complete your presentation, you can go back to your home before the election. Okay, so I think it is appropriate. So your exam will be on 19th, then you will submit your assignment on 23rd. Actually, when you are preparing your assignment at the same time, already you know your group, you will start working with your presentation and just give the presentation on 26th. And in the presentation, you are not going to present the whole thing. Only each group are having 4 amino acids. That is not a huge job. This is just a few portion of your assignment, and you should be able to do that. Actually, when you are preparing your assignment, then you can prepare your PowerPoint same time. It's the same topic. Is it clear?
Yes ম্যাম।
Okay, so please note down the dates And my target is I have to complete after today's lecture 4 more lectures. And actually among those 4 lectures, 2 lectures are very small and other 2 lectures are quite like huge content. So I hope I can complete it. But another one, we have to do one more quiz. And that quiz, I want to fix it on 9th December. 9th December, I will book a room for your quiz. Hopefully about 10:10 or maybe 11:50. Please write it down. Write it down, and everything you should post in the classroom. Otherwise, I will mix your schedule with another course's schedule. Okay. So, like because we have completed 4 quizzes, is not it? And one more quiz I want to
Yes,
take,
ma'am.
and that will be on 9th December. And the time, now at this moment I cannot confirm it, maybe 10:10 or 11:50. I'll let you know by Saturday. Sorry, Sunday or Saturday. And then our final exam is on 19th. Then your assignment submission on 23rd and your presentation on 26th. Okay. So whatever like lecture or what is the topic we complete in this remaining 4 classes after we have done our mid, that will be our final content, okay, for the exam. Is it clear? Do you have any confusion? Everyone?
No, ma'am.
Okay. Okay, so let us start with our today's lecture. Can you see it?
Yes, ma'am.
Hello,
Yes,
is
ma'am.
it visible? Okay, so from today we are going to start the anabolic phase of metabolism. And we already know that anabolism means where a molecule is synthesized from simple precursor molecules, and these synthesis reactions, they need energy, and there are 2 forms of energy. One is the ATP, another one is the reducing power, that is NADH or NADPH. And these reactions or anabolic reactions are actually reductive, whereas catabolic reactions were oxidative. And in the living organism or cells, these anabolic reactions actually happen simultaneously with catabolism. Okay, so it is not that at first anabolism happens, then nothing happens within the cell, and when it requires, then the catabolism will happen. Obviously, that is actually determined by the requirement of energy within the cell, as well as cell also need to counterbalance in between the anabolic phase and metabolic phase of the metabolism. Okay, so actually from this lecture we are going to start the biosynthesis of the macromolecular precursors. That means we have already learned the macromolecules and what are their precursors. So mainly here we are focusing on the biosynthesis of the macromolecular precursors. So at the very first one, we are focusing on the biosynthesis of carbohydrates. So the precursor of carbohydrates are the simple sugars. So that is our topic. So how the living organism synthesize the simple sugars. And the thing we have actually learned from our school that the living organisms which have chlorophyll, those are the plants and other microbes. they can do photosynthesis, where they can actually synthesize glucose from carbon dioxide and water, and obviously the energy ATP and the reducing power NADPH are used in this photosynthesis. And that reaction is also light dependent. That means light energy is required. So plants and other photosynthetic microbes, they can do that, but animals, they cannot do this reaction. So animals also need the macromolecule carbohydrates. So how the animals get the simple sugars? Obviously, the simple sugars are coming from their food, which is, or that means maybe triose or hexose. Those are coming from the plants, and then the animals, they can synthesize other macromolecules or carbohydrates, and the simple precursor in animal cells, that is the 3-carbon precursor. Okay, that is the simple precursor of carbohydrate in animal cells, 3-carbon precursors. And that obviously less oxidized than the carbon dioxide. Okay, that is the main difference in between plants and photosynthetic organism with the animals. Okay, now we know that plants are autotrophs, so they have the ability to use carbon dioxide as the sole source of carbon, and from there they get the precursor molecules, which they actually utilize to synthesize all the other macromolecules, whether it is different types of carbohydrates, then lipids and proteins. Okay, so now we will focus on the carbon dioxide, because carbon dioxide is present in the environment. So this is done by the organelle, chloroplast, and chloroplasts are present in the green plants, and chloroplasts actually have specific enzymes, and those enzymes catalyze the conversion of carbon dioxide to simple organic compounds, which we said that carbon dioxide is reduced to organic compounds, and this process is called carbon dioxide assimilation, and it is also known as carbon dioxide fixation or carbon fixation. And this carbon dioxide is actually incorporated into a 3-carbon compound, 3-carbon organic compound. Obviously, there is a carbon compound, and that is a triose. And that triose is phosphate 3 phosphoglycerate. Okay, sorry, triose phosphate. That means a 3-carbon sugar together with a phosphate group, and that sugar is 3-phosphoglycerate. So this is the like organic compound which is generated by the fixation of carbon dioxide, and from this precursor, that is the 3-phosphoglycerate, now it is used to synthesize other sugars or the polysaccharides, even other metabolites. Okay, even
Miss,
the rest of the pathways, that is from the 3-carbon sugar, whereas other sugars or other biomolecules are synthesized, all those pathways are almost same in animal and plant cells. Okay, but the fixation part only occurring in plants or the microbes which have chlorophyll. And this pathway by which the carbon dioxide is fixed, that happens in a cyclic pathway. Okay, it is a cyclic pathway. And here, so obviously we learned cyclic pathway already, TCA cycle. So in this cyclic pathway, the intermediates are regenerated, and this pathway is called Kelvin cycle, or more elaborate name is photosynthetic carbon reduction cycle, because obviously in this pathway the carbon dioxide is reduced. That's why it is known as photosynthetic carbon reduction cycle. Is it clear?
Miss,
Yes, ma'am.
Yes.
অর্গানিক কম্পাউন্ডটার নাম কি ট্রায়োস ফসফেট 3-ফসফোগ্লিসারেট নাকি শুধু 3-ফসফোগ্লিসারেট?
3-phosphoglycerate.
মানে
The
ওইটা ট্রায়োস ফসফেট
general name is triose phosphate. That means it is a triose sugar
বুঝতে পারছি।
made up of 3 carbons. Clear?
ইয়েস মিস।
Okay. So in this reaction, energy is required. So from where the energy is coming, and the two forms of energy, one is ATP and another one is NADPH, and these two forms of energy is actually synthesized by light dependent reactions of photosynthesis. Okay, so if you look at the slide here, the light, the energy is consumed, and that energy is synthesized to it used to synthesize ATP and NADPH, and the carbon dioxide and water, those are used to synthesize at first the triose phosphate. Then eventually the triose phosphate will be converted into hexose phosphates, and hexose phosphates, which are actually monomers of different carbohydrates, such as it is used to synthesize the disaccharide. Then it is used to synthesize starch, which is the polysaccharide, or other components of the plant, such as cellulose. And we already know hexose are also used to synthesize the pentose sugar, and we know that pentose sugars are the component or part of the nucleic acid. So they are utilized to synthesize the nucleic acids, or from other metabolites, they are used to synthesize the other macromolecules, protein or lipid. Okay, so this is the thing happening specifically in plants. So plants are the one which actually converting the inorganic compound or inorganic carbon into organic compound. Okay, so the most important organelle where the carbon dioxide fixation is occurring is called, general name is plastid. So they are self-reproducing organelles. It has same features as mitochondria. We have learned in the previous lecture. They have double membrane. They also have genomes. So some of the genes of the genomes encodes their protein, and the biosynthetic activities, that means the anabolic reactions or pathways mainly involving the carbon dioxide fixation happened in plastids. There are different types of plastids. Obviously the differences are about their the presence of the pigment, or the what is the feature of the inner membrane. The first one is the chloroplast, where carbon dioxide assimilation occurs, and that means all the enzymes which are involved in this method, they are present in the stroma of the matrix, of the chloroplast. So stroma is the matrix, or we can say it is the same homologous location as mitochondrial matrix. Second one is amyloplast. Amyloplast are colorless plastids. So chloroplast, they have the pigment chlorophyll, whereas amyloplast, they don't have chlorophyll or any other pigment, as well as they don't have internal membranes, which are like the photosynthetic membranes known as thylakoids. So they don't have these. And what is their main function? They actually filled with starch granules. That means after the starch is synthesized, they are stored within the amyloplast. Third one is proplastids. They are actually converted or coming from the chloroplast, when the chloroplast lost their internal membranes and chlorophyll. So that is actually, at first there is chloroplast, eventually the chloroplast lost their chlorophyll and the internal membranes, and then they become proplastids. Eventually the proplastids are becoming amyloplast. And the main function of the amyloplast is to store carbohydrate. So if we look at the green plant or the plant, we can now easily say that the green leaves of the plant, they are actually rich in the chloroplast because they have the chlorophyll. And amyloplast, they are the non-photosynthetic tissues. That means photosynthesis does not occur in amyloplast. Rather than they store the starch in large quantities. An example, potato tubers. So potato tubers are made up of amyloplast. Is it clear?
Yes, ma'am.
Yes, ma'am.
Okay. Maybe some of you already completed 301. So you may like have or already have the knowledge of this pathway. But that is the topic of biochemistry, metabolism. Okay, now the particular pathway, which is the pathway where carbon dioxide is assimilated or fixed. And that carbon dioxide fixation occurs in 3 stages, mainly 3 stages. And in this assimilation pathway, the carbon dioxide is accepted or condensed with an acceptor molecule, which is a sugar. So this is the 5-carbon sugar, ribulose 1,5-bisphosphate. So this is a ribulose, means what type of sugar it is? Ribulose 1,5-bisphosphate.
Pentose sugar,
Hmm?
ma'am. Pentose sugar.
Pentose, ribose sugar.
Ketose. Yes, ketose. This is the ketose, and we can clearly see it has the keto group at carbon number 2. So this is the acceptor molecule of carbon dioxide, and when the carbon dioxide condensed with it, obviously an enzyme is doing this reaction, and the name of the enzyme is, according to the acceptor molecule, ribulose 1,5-bisphosphate carboxylase and oxygenase, oxygenase. So that means this enzyme has 2 activities. One is carboxylase activity, another one is oxygenase activity. So that is the fixation step. And after that, the 3-carbon compound is generated, which is 3-phosphoglycerate. So you can see the 3-phosphoglycerate is produced. Okay, now the 3-phosphoglycerate undergoes at first phosphorylation. So if you look at this reaction, phosphorylation, that means ATP is required in that reaction. And the enzyme which is synthesizing this reaction is called 3-phosphoglycerate kinase. Kinase is the enzyme doing phosphorylation. And we get 1,3-bisphosphoglycerate. Okay, so because this intermediate, it has phosphate group at the carbon number 3. And now one more phosphate group is added at the carbon number 1. So that compound, which is not shown here, but that is also important intermediate in this pathway, that is 1,3-bisphosphoglycerate, which actually eventually undergo reduction reaction by the enzyme glyceraldehyde 3-phosphate dehydrogenase, according to the name of the product here. You can see ultimately after the reduction reaction, glyceraldehyde 3-phosphate is produced. Okay, and it is a reduction reaction, and you can see the reduced power is provided by the NADPH. Okay, so 1,3-bisphosphoglycerate is reduced to glyceraldehyde 3-phosphate. Okay, now this is the actually ultimate a 3-carbon sugar which is produced in this cycle, and actually majority of the glyceraldehyde 3-phosphate is used to regenerate the acceptor molecule, which is ribulose 1,5-bisphosphate, and that reaction also requires ATP, because ribulose bisphosphate 1,5-bisphosphate has 2 phosphate groups. Only one of the glycylate triphosphate that actually used different purposes, obviously depending on the demand of the cell. It can be used in the glycolysis pathway, or maybe it can be used to synthesize other sugars, that means hexoses or the polysaccharides. So this is a 3-carbon sugar. So from here, the different hexoses can be produced, whether it is an aldose or ketose. So according to that, the particular enzyme will be used, such as maybe aldoses or transketoses, which are the specific enzymes responsible for the synthesis of ketose. So ultimately, this is the cycle where the inorganic carbon as carbon dioxide is fixed into organic compound. Is it clear?
Yes, ma'am. Yes, ma'am.
So here is the description of this reaction or pathway, and this is the stoichiometry. That means how many molecules of the acceptor molecule and the carbon dioxide, and how many molecules of the intermediates are produced in this cycle. So you can see 3 molecules of ribulose 1,5-bisphosphate condensed with 3 molecules of carbon dioxide, and we get 6 molecules of 3-phosphoglycerate, and from there that is phosphorylated by the enzyme 3-phosphoglycerate kinase, and we get 6 molecules of 1,3-bisphosphoglycerate, which is then reduced to the 3 carbon sugars here. Actually, 2 sugars are produced, glycerol 3-phosphate and dihydroxyacetone phosphate. And if you remember, we learned about that because they are isomers. One is aldose sugar, another one is ketose sugar. So among these 6, only one of the sugar, triose sugar, that is actually utilized for energy production or synthesis of other sugars or the polysaccharides, whereas 5 molecules of the 3-carbon sugars, they are utilized to synthesize the acceptor molecule. So if you look at this reaction, they are condensed. Actually, they are undergoing the condensation reaction and give rise to the 5-carbon ketose, ribulose 5-phosphate, which is then phosphorylated into 3 molecules of ribulose 1,5-bisphosphate that is ready to condense with 3 molecules of carbon dioxide. Okay, now next one that is very important, which is called photorespiration. So in photorespiration, it is like it has similarity with the cellular respiration, which occurs in mitochondria, and where we have seen carbon dioxide is used and oxygen is used and carbon dioxide is produced, and that reaction is done in presence of light. And in this case, it is also same, and in this case, it is happening in the presence of light, and the oxygen is utilized and carbon dioxide is produced. But this is a costly side reaction of photosynthesis. And why this is happening? Because the enzyme, which is the ribulose 1,5-bisphosphate carboxylase or oxygenase, the short name of the enzyme is Rubisco, and this enzyme, it is not specific for its substrate. Actually, the enzyme Rubisco, it has 2 substrates. One is carbon dioxide and another one is oxygen. So these 2 substrates competes for the active site. So which substrate will bind to the active site of the enzyme? And the substrate which actually binds to the active site, according to that, the reaction will happen. If the substrate is oxygen, then the oxygenase activity of the enzyme will do the reaction. Okay. And if the substrate is carbon dioxide, then the carboxylase activity of the enzyme will do the reaction. So when it is carbon dioxide, that reaction we already learned. In case of carbon dioxide, the carboxylase activity of the enzyme will do the reaction and we get the 3-phosphoglycerate. But if the oxygen is bound to the active site, then the oxygenase activity of the enzyme will do the reaction, and then actually we get two products. One is 3-phosphoglycerate like the previous one, and one more product which is 2-phosphoglycolate. Okay, so this 2-phosphoglycolate, which is actually metabolically useless product. So we already know the 3-phosphoglycerate, which is actually ultimate precursor for other sugars or the polysaccharides and eventually other biomolecules. But the 2-phosphoglycolate, which is produced by the oxygenase activity of Rubisco, that is metabolically useless. That means the cell kind of cannot utilize it. But cell has a way to use it, and that is called the salvage of the phosphoglycolate, and that is very costly for the cell. Actually, the glycolate pathway is the one by which this phosphoglycolate is converted to the amino acid serine and a molecule of carbon dioxide. But that is very costly for the cell. So now the glycolate pathway, and the glycolate pathway is actually occurring in 3 organelles.
মিস কথা শোনা যাচ্ছে না।
So this 2-phosphoglycolate is dephosphorylated by releasing the phosphate group. It produces glycolate. So the structure is also shown here, the chemical structure. You can see glycolate, it has 2 carbons. At number 1, it has the carboxylic group, and another carbon, it has the hydroxylic group. That glycolate then
মিস একটু আগে কথা
exported
শোনা
to
যায়
the
নাই। মিস একটু আগে আপনি
fire
মিস ওই যে আপনি শুরু
which
করলেন গ্লাইকোলেট পাথওয়ে থেকে, তখন থেকে মানে কথা স্টপ হয়ে গিয়েছিল, কথা শুনি নেই।
okay. So glycolate pathway occurs in 3 organelles of the cell, and they are chloroplast, paroxysm, mitochondria. So in the chloroplast, the Kelvin cycle happens, and instead of carbon dioxide, here the ribulose 1,5-bis phosphate condensed with oxygen. As usual, the 3-phosphoglycerate, which is produced, that can actually run the Kelvin cycle, but the another product, which is the 2-phosphoglycolate, that cannot enter Kelvin cycle because it cannot be utilized here. So this 2-phosphoglycolate undergoes dephosphorylation and we get glycolate. And if you look at the structure of the glycolate, it has a carboxylic group and a hydroxyl group. Now this glycolate transported to the next organelle, which is paroxysm. Within the paroxysm, the glycolate is oxidized. Okay, that means another oxygen is required in this reaction. Glycolate is oxidized by the enzyme glycolic acid oxidase. Okay, so the molecular oxygen is directly participating in this reaction. Glycolate becomes glyoxylate. Okay, glyoxylate. So this is the oxidation reaction. This hydroxyl group is becoming the acetaldehyde group, aldehyde group. You can look at the structure, chemical structure, carboxylic group and aldehyde group, that is the glyoxylate. Now the glyoxylate undergoes transamination reaction. transamination reaction. We have already learned it during the amino acid catabolism, and glyoxylate actually becoming the amino acid glycine. Okay, glycine, that means this oxygen that is actually displaced with an amino group and becomes glycine. Now the amino acid glycine goes to the next organelle, that is mitochondria, where two molecules of glycine condensation reaction, and that condensation reaction is catalyzed by glycine decarboxylase, and decarboxylase that is doing or releasing carbon dioxide. So carbon dioxide is released here. Okay, that is the carbon dioxide which is released here, and the glycine becomes the amino acid serine. Okay, serine. And this reaction requires the participation of the electron acceptor NAD plus. Okay, and carbon dioxide is released here. So this carbon dioxide that can actually condense to it in the Kelvin cycle. Okay, now the serine, the amino acid we get in the mitochondria after condensation of two molecules of glycine, the serine is transported to the peroxisome. So here serine is undergoing the transamination reaction, that is the amino group of serine is actually here transported or given to the glyoxylate, which is becoming glycine. And after donating the amino group, serine is becoming hydroxypyruvate. Okay, after removing the amino group, you can look at the reaction. That amino group is removed, and that is replaced by a keto group, and that produces hydroxypyruvate. Okay, now this hydroxypyruvate, so if you remember, pyruvate, it has 3 carbons, but it has a hydroxyl group at carbon number 3, hydroxypyruvate. So this hydroxypyruvate undergoes reduction reaction. Reduction, that means where the keto group is reduced into hydroxyl group, and the reduction electrons are provided by NADH, and it becomes glycerate. Okay, glycerate. Glycerol is made up of 3 carbons. Okay, here the 2 carbons, they are having the hydroxyl group, and another one is the carboxylic group. That's why it is glycerate. Now this glycerate is transported to the chloroplast, where the glycerate is phosphorylated. Okay, it is phosphorylated. So that means once it is phosphorylated at the carbon number 3, it becomes the 3-phosphoglycerate, and that 3-phosphoglycerate can enter the Kelvin cycle. So like, to do this, you can see the cell actually needs to do many steps, and those steps are occurring in different organelles of the cell, and obviously at the same time many reducing powers as well as ATP is required. So it is very costly for the cell. বোঝা গিয়েছে?
Yes, miss.
So that is the glycolate pathway. Okay, so actually we can say that because this is the very fast mechanism or the pathway by which the plants fix the carbon dioxide into a 3-carbon compound, that is the 3-phosphoglycerate. And because of that, the plants which can fix the carbon dioxide by this pathway, they are known as C3 plants. Is it clear? The plants which fix carbon dioxide by this pathway, they are known as C3 plants. So
Yes, ma'am.
মিস, যারা অক্সিজেন বাইন্ড করে তাদেরকে তাহলে কি বলে?
অক্সিজেন
মানে
বাইন্ড করে মানে কি?
মানে কার্বন
অক্সিজেন তো সব সময় বাইন্ড করবে না। The major goal of this pathway is to fix carbon dioxide, not oxygen. But oxygen is competing. Oxygen is competing with carbon dioxide. So if oxygen gets the opportunity, then this will happen. But
মানে মিস যখন অপরচুনিটিটা পাবে অক্সিজেন তখনই মানে ওটাকে কি তখন মানে অন্য কোনো স্পেসিফিক নেম আছে কিনা ওটা সেটা বললাম।
আমি বুঝলাম না, সেটাই তো গ্লাইকোলেট পাথওয়ে। যদি অক্সিজেন বাইন্ড করে তখন তো এই সিচুয়েশন হবে, আর অক্সিজেন বাইন্ড না করলে তো দ্য সিম্পল কেলভিন সাইকেল।
আচ্ছা মিস ওটাকেই C3 বলে তাই তো?
Obviously,
ওকে
that
মিস।
is the C3. And if the oxygen wins the competition, then this glycolate pathway will go on, because that glycolate or 2-phosphoglycolate which is produced, that cannot be utilized unless you convert it into a usable product.
বুঝতে পেরেছি।
Clear to all of you?
Yes, ma'am.
Is it clear in which situation the plant is doing this? That is not the regular pathway. If oxygen condensed with ribulose 1,5-bisphosphate instead of carbon dioxide, this will happen. But the original pathway where the carbon dioxide is fixed as the 3-carbon compound, 3-phosphoglycerate, and the plants which can do that, they are known as C3 plants. Okay, now it is not obviously necessary that all plants they fix the carbon dioxide as the 3-carbon compound, 3-phosphoglycerate. There are plants which can fix the carbon dioxide as a 4-carbon compound. Okay, so obviously they will be called C4 plants. So those plants, actually, what are the examples of those plants? They actually grow in the tropics and the temperate zone crop plants. Examples are maize, sugarcane, and sorghum. Those are the plants which can fix the carbon dioxide as 4-carbon compound. That's okay. Now look at this pathway, how this fixation is happening. So in this pathway, the carbon dioxide actually does not fixed or does not condense with the acceptor molecule. At first, the carbon dioxide present in the environment, that is becoming bicarbonate by interaction or reaction with water. Now this bicarbonate is condensed with the acceptor molecule, and here the acceptor molecule is totally different from the C3 plants. Here the acceptor molecule is phosphoenol pyruvate, and that fixation happens in the mesophyll cell of the plant, C4 plants, by the enzyme phosphoenol pyruvate carboxylase, and it fixes the carbon dioxide as oxaloacetate. Oxaloacetate is the 4-carbon compound here. Okay, oxaloacetate. Now the oxaloacetate, which is reduced to malate by the enzyme malate dehydrogenase. Now this malate, which actually can be, there is a possibility malate can be oxaloacetate can be reduced to malate, or maybe oxaloacetate undergoes transamination reaction to the amino acid asparagine. তাই না? আমরা কি শিখেছিলাম ওই যে ইউরিয়া সাইকেলে oxaloacetate undergoes transamination reaction, and what we get? মনে আছে কারো? Asparagine. So that can happen. It depends on the, again, what is the requirement of the cell. Now oxaloacetate in this, because here we are focusing the fixation of carbon dioxide. So this malate that will go to the bundle sheath cell by a transmembrane protein which is known as plasmodesmata. This is a like a protein channel through which small molecules or small proteins can be transported in between two cells, and that will be transported to the bundle sheath cell. Within the bundle sheath cell, this malate is oxidized. Okay, malate is oxidized by the enzyme, malic enzyme, and when this oxidation happens, carbon dioxide is released and obviously electrons are accepted by the NADP plus, and we get pyruvate. This is the carbon dioxide. If you look at the figure, the carbon dioxide, which is actually fixed at the very beginning, and that carbon dioxide is coming from the environment, and now it is released in the bundle sheath cell, and this carbon dioxide then condensed with here the actual reaction we learned in the Kelvin cycle, ribulose 1,5 bisphosphate, and becomes the 3 phosphoglycerate, the 3 carbon compound. Okay, do you get it? The actual Kelvin cycle is happening here, but the carbon dioxide here, it is fixed in a different way, and then eventually that carbon dioxide is used in the Kelvin cycle. Do you get it? How it is happening?
Yes, ma'am.
Okay, now we get the pyruvate by the action of the Malick enzyme. So pyruvate, it goes to the mesophyll cell by the plasmodesmata, and pyruvate then undergoes phosphorylation reaction by the enzyme pyruvate phosphate dikinase, because 2 phosphate groups are added to the pyruvate, and you can already see ATP is like degraded into AMP and inorganic pyrophosphate, and we get phosphoenol pyruvate, which is ready to accept another molecule of bicarbonate. Okay, so that is the carbon dioxide fixation pathway happening in C4 plants. So the description is here. Okay, now there is another group of plants which are called CAM plants. Actually, they have specific features. They are also known as succulent plants, and examples of such plants are cactus, pineapple. So actually, if you look at these kind of plants, their leaf and stem is very thick, and also they have thorn-like structures, and they have a variation about the carbon dioxide fixation, actually variation from the C4 plants. Okay, C4 plants which fix the carbon dioxide as the 4 carbon compound, they are actually utilizing that particular pathway, but in a different way, because this kind of plants usually they grow in a very dry environment, especially the desert. So what happened in the desert at the daytime, the temperature is very high and the environment is very hot, and during that situation, because the plants they have the stomata, usually that stomata is used to get the carbon dioxide and oxygen to the leaf tissue. But if that is open during day, obviously the water will be lost from the plant, and that is not convenient for the plant survival. So what these kind of plants do, they keep the stomata closed during daytime. So that means if it is closed during the daytime, then the carbon dioxide and water cannot enter the leaf tissue. So, but plants need it. So when the plants are going to um, get the carbon dioxide, specifically here it is about carbon dioxide. So carbon dioxide will be used or like consumed during the night when the environment is cool and moisture, that means water is present in the environment during night. At that time, the stomata will be open through which the carbon dioxide will enter the cell, and the rest of the reaction will happen like the C4 plants. That means the carbon dioxide at first it will be converted into bicarbonate, and then it is fixed into oxaloacetate by phosphoenol pyruvate carboxylase. Okay, now in the next step, we have seen that the oxaloacetate is reduced to malate, but during the night, this malate is stored in the vacuoles. Okay, they are stored in the vacuoles. They are not released. Why they are not released? Because in that situation, if they are released, they actually can interfere with the chemical environment of the cell. That means they can interfere with the cytosolic and plastid enzymes by the low pH, which actually generated by the malic acid dissociation reaction. So during the night, they don't do that. They keep the malate within vacuoles. Now, when it is daytime, the stomata, at that time, the stomata is closed. Because, when night, the water may go out of the cell. But at the daytime, the stomata is closed. There is no loss of water. And then the malate, which is stored within the vacuoles, they will be released as by the action of the malic enzyme as the carbon dioxide. Carbon dioxide then fixed by the Kelvin cycle as 3-phosphoglycerate. Okay, so the rest of the reactions are same. It's only the fixation or like the steps are happening at different time points for this group of plants. And this particular fixation pathway was first discovered in particular species of the plant, which are stone crops, perennial flowering plants of the family Crassulaceae. Crassulaceae is the family of the plant, and that's why it is known as Crassulacean acid metabolism. And the abbreviation is CAM plants. We get it?
Yes, ma'am.
Is it clear?
Yes, ma'am.
Okay, so the carbon dioxide now fixed by different group of plants in different ways, but all the plants, they are actually utilizing the Kelvin cycle. Okay, Kelvin cycle. Okay, now we get the triose. So from that triose sugar, the cell now synthesize like hexose, and from hexose, the disaccharide and eventually polysaccharides are synthesized. So we will learn some of them. The first one is the biosynthesis of starch. Okay, here actually slideshow. Let me know whether you can see it. Can you see it? Can you see the slideshow?
Yes,
No,
ma'am.
ma'am. আগের মতোই
Not
আছে
yet.
তো ম্যাম।
ম্যাম আগের মতোই আছে।
But you are seeing the slide biosynthesis of starch.
Yes, ma'am.
Yes, ma'am.
Okay. So starch, we already know this is a polysaccharide, and it is present in the plant, and it is made up of D-glucose, and the glucose residues are linked by alpha-1,4 glycosidic bond. And starch is synthesized in the chloroplast. That is the actually stable end product of photosynthesis. Okay, what is the precursor here? Glucose. So actually, how the glucose is delivered during the biosynthesis of starch, sugar. It is not that glucose is coming and that is added to the chain. So the glucose is delivered as activated nucleotide sugar. That means glucose is added to a nucleotide, and that nucleotide is ADP. Okay, and the nucleotide sugar is called ADP glucose. And from where we get ADP glucose? That ADP glucose is synthesized from the reaction in between glucose 1-phosphate and ATP. Okay, glucose 1-phosphate and ATP interacts and we get ADP glucose, and this is an irreversible reaction which occurs in the plastids, and the enzyme which is doing the reaction is called inorganic pyrophosphatase. Okay, the phosphate group is removed from the glucose 1 phosphate, and there it actually the ADP is added to that glucose. Enzyme is inorganic pyrophosphatase. Then this ADP glucose is delivered to the enzyme, and that enzyme is starch synthase. Okay, so ADP glucose is providing the precursor, which is the glucose, to the enzyme starch synthase, and starch synthase then add these glucose residues to the pre-existing starch molecule. Okay, now we look at the reaction. If you look at the reaction here, the enzyme starch synthase, which has, you can see, two active sites, XA and XB. At the very beginning, there comes two ADP glucose residues, and those glucose residues number 1 carbon is attacked by the active sites, two active sites of starch synthase, and that attack happened because of the nucleophile attack. Okay, the active sites, they are rich in electron and they attack the number 1 carbon of ADP glucose, which is here functioning as electrophile, and because of that it gives rise to the bond. That means the active site interacts with the carbon number 1 of the sugar residue. Okay, now in the second step, what happens? If you look at the figure, the sugar residue 2, which is attached to the active site XB, and sugar residue 1 attached to the active site XA. So the number 2 sugars hydroxyl group at carbon number 4. So this hydroxyl group is functioning as a nucleophile and attacks the number 1 carbon of the sugar 1. Okay, so when this reaction is happening, so sugar 1 will be released from the active site XA, and that will be linked to the sugar 2 by alpha 1,4 glycosidic bond, which causes the XA site free. Now the XA site is free, so another ADP sugar delivering the sugar residue to the XA. So same thing will happen. XA interacts with that number 3 sugars, number 1 carbon. And now in return, we can see the 2 sugars, number 1 and 2, they are linked to the XB side, and number 3 sugar is linked to the XA side. Now the number 3 sugars hydroxyl group is functioning as the nucleophile, attacking the number 2 sugars, number 1 carbon. Okay, by that the glycosidic bond 1,4 glycosidic bond is produced in between the number 2 and number 3 sugar residues. Okay, so this is the way it will go on, which will add the sugar residues one by one, and that will elongate the starch polymer. Okay, by the enzyme starch synthase. Okay, now is it clear to you? কিভাবে রিঅ্যাকশনটা হচ্ছে আমরা কি বুঝতে পেরেছি?
Yes, miss.
Okay, so that is the biosynthesis of starch or a polysaccharide. Okay, next one. Is it move to the next
নো ম্যাম। ম্যাম আপনার
ম্যাম,
কথা
আপনার
শোনা
কথা
যাচ্ছে না।
শোনা
এখন
যাচ্ছে
বেঁধে,
না।
বেঁধে আসছে।
ম্যাম কি লিভ নিয়েছেন?
জানি না, মনে হয় তো। স্ক্রিন কিছু দেখা যাচ্ছে না।
ম্যামের কারেন্ট গেছে।
Sorry, I was disconnected. Okay, can you see it now?
ও ম্যাম।
নো ম্যাম।
The slide.
ইয়েস ম্যাম।
Yes, ma'am.
ইয়েস ম্যাম।
So we have completed the starch biosynthesis. Do you have any question regarding that starch biosynthesis? Hmm? Any
নো ম্যাম।
question about starch? Okay. So if you don't have any question, then we will look at the biosynthesis of a disaccharide. For example, it is sucrose. Sucrose is a disaccharide, we know that, and it is made up of the sugars, glucose, and fructose. So, what are the precursors of like sucrose? At first, we will say glucose and fructose, that those are the monomers. So, from where we get glucose and fructose? Okay, so that actually, or we can say the very simple precursors of sucrose is the triose, which are dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. And we know that from where we get it, কে বলতে পারবে? এখানে বলছে they are exported from chloroplast. So from where we get dihydroxyacetone phosphate and glyceraldehyde 3-phosphate in chloroplast? কে বলবে? Hmm? In the chloroplast, how we get dihydroxyacetone phosphate and glyceraldehyde 3-phosphate? কে বলবে? আমরা কি বুঝতে পারছি? Hello,
ইয়েস মিস।
I am asking you something. So আমি কি বলছি বলো? এই যে ডাইহাইড্রক্সিঅ্যাসিটোন ফসফেট এন্ড গ্লিসালডিহাইড 3-ফসফেট, এটা ক্লোরোপ্লাস্টে কিভাবে আসলো?
ম্যাম কেলভিন সাইকেল থেকে।
ক্যালভিন।
এত সময় লাগে কেন?
সরি ম্যাম,
আজকের
কানেকশন
টপিকটাই
প্রবলেম
তো
হচ্ছিল।
এইটাই, মানে আজকের টপিকও যদি না বলতে পারো তাহলে কেমনে?
মিস
ওকে,
এখানে মাঝে মাঝে কানেকশনের প্রবলেম করতেছে, সাউন্ড মানে আপনার কথা কেটে কেটে যাচ্ছে।
okay, sorry about that. So we get the dihydroxyacetone phosphate and the glyceraldehyde 3-phosphate, the trio sugars by the Kelvin cycle. Okay, now these two sugars, they condensed. কি রিয়াকশন কোথায় পড়েছো এটা? এই দুইটা ট্রায়োস সুগার, they condensed and give rise to a hexose, নাকি? কি রিয়াকশন বলে এটাকে?
গ্লাইকোলাইসিস।
গ্লাইকোলাইসিস, হ্যাঁ বলতে পারি। গ্লাইকোলাইসিসে আমরা তো দিস ইজ এ রিভার্সিবল রিয়াকশন, তাই না?
ইয়েস ম্যাম।
ইয়েস ম্যাম।
বাট যখন আমরা ওই যে বায়োকেমিক্যাল রিয়াকশন শিখলাম তখন আমরা শিখিনি কার্বন কার্বন বন্ড মেকিং এন্ড ব্রেকিং, মনে পড়ে?
ইয়েস ম্যাম। ইয়েস
মিস
ম্যাম।
অ্যালডোল
Yes.
কন্ডেনসেশন।
Aldol condensation catalyzed by aldolase. So these two trios, they are condensed and give rise to a hexose, which is fructose 1,6-bisphosphate. Now this fructose 1,6-bisphosphate, it is hydrolyzed by the enzyme fructose 1,6-bisphosphatase. Okay, fructose 1,6-bisphosphatase according to the name of the substrate. Then we get fructose 6-phosphate. So here the fructose 6-phosphate is shown in this reaction. You can see fructose 6-phosphate, but how we get fructose 6-phosphate, that is not shown in this slide. But I want you people, you have to also show that reaction, how we get fructose 6-phosphate. We get fructose 6-phosphate from the two trios which are condensed. At first we get fructose 1,6-bisphosphate that undergoes hydrolysis by the enzyme fructose 1,6-bisphosphatase, which removes the phosphate group from the carbon number 1, and we get fructose 6-phosphate. So this is one of the precursor in the biosynthesis of sucrose, and another one is glucose. So glucose, again, it is not just the single glucose or simple glucose. The glucose need to be activated, like we have seen in case of starch synthesis where the glucose is activated by ATP. So here the glucose is activated by the nucleotide UTP, and it produces UDP glucose, and UDP glucose then combined or condensed with fructose 6-phosphate. In that condensation reaction, UDP will be released, and you can see the bond, glycosidic bond, which is generated in between the carbon number 1 of glucose and the carbon number 2 of fructose by the enzyme sucrose 6-phosphate synthase. And we get sucrose 6-phosphate. Next step, we have to remove this phosphate group from the sucrose 6, carbon number 6. And that is done by the enzyme sucrose 6-phosphate phosphatase. Phosphatase is the enzyme which removes the phosphate group. And after that reaction, we get the disaccharide sucrose. Glucose and fructose, they are linked by 1,2 glycosidic bond. Is sucrose reducing or non-reducing?
Non-reducing ম্যাম।
Why?
বিকজ ম্যাম এদের OH গ্রুপটা হচ্ছে ব্লক হয়েছে।
কোন OH? শুধু OH গ্রুপ বললে হবে?
ম্যাম
না
অ্যানোমেরিক
ম্যাম, ওই যে অ্যানোমেরিক।
কার্বনের যে OH।
Yes. So anomeric যদি না বলো, your answer is wrong. Okay, তাহলে বুঝলাম আমরা biosynthesis of disaccharide. Then again a polysaccharide, specifically cellulose, and that is the component of the plant cell wall. Actually, this is not the actual pathway. This is just a model proposed by the scientist. So they actually observed the plant cell membrane under electron microscope, and they found a rosette-like structure, that means flower-like structure, which is made up of 6 units, and each of the unit is actually the enzyme cellulose synthase. So cellulose synthase is the enzyme which is doing the biosynthesis of the polysaccharide cellulose. And here we know that the precursor is the glucose, and the glucose are linked to each other by beta 1,4 glycosidic bond. And the way we have learned in case of biosynthesis of starch and sucrose, that the glucose is delivered by some other molecules, that means glucose need to be activated. So here as well, if you look at the model, the sucrose synthase is present in the plasma membrane of the plant cell, and the glucose residues are delivered by different glucose carrier molecules such as sterol. Sterols are present usually in the membrane. So specific one here, cytosterol, beta glucoside, which is delivering the beta glucose residues, or the glucose can be synthesized from sucrose, and glucose will be delivered as UDP glucose, and then the cellulose synthase can utilize that glucose to synthesize the cellulose. So the glucose is added to the chain, and ultimately it is actually elongated outside of the plasma membrane. So that is actually the model, and you can also see the monomer of the glucose synthase. So that means this is just the simple structure of the enzyme glucose synthase according to the protein structure. This is a globular protein. If you look at the structure, it has amino end, carboxylic end, which are facing the cytoplasm, and they have the membrane spanning region, and there is also some part which are present within the cytoplasm, and they are attached to some sugar residues. So we say that glycosyl transferase motifs of the protein. So they are responsible for the transferase of the sugar residues. So this is just simply a model. The scientists they like generated the model. Maybe this is the way how the cell is synthesizing the cell wall made up of glucose from sugar residues. Okay, so that is about our today's lecture. Any question?
মিস হচ্ছে গত যে লেকচারটা ছিল ওটা তো লাস্টে 2টা স্লাইড তো মিস পড়ানো হয়েছিল না। মডেল পর্যন্ত পড়ানো হয়েছিল ক্লাসে। রেগুলেশন অফ অক্সিডেটিভ ফসফোরাইলেশন এটা।
ওটা তো ইন জেনারেল তো বলেই দিছিলাম মনে হয়। বলেছিলাম না যে demand of energy which is determined by the concentration of ATP and ADP and inorganic phosphate.
Yes, ma'am.
And all the interaction of the other pathways, because different pathways they are interlinked. বলেছিলাম তো বোধহয়।
Yes, ma'am. বলেছেন।
Okay, any question?
হচ্ছে আমাদের 9 তারিখের যে এক্সামটা এটা ম্যাম আমাদের ক্লাস টাইমে নেওয়া যায় না? আমাদের অনেকেরই ম্যাম হচ্ছে কেমিস্ট্রি ল্যাব এক্সাম আছে 9টা থেকে 11টা পর্যন্ত।
তাহলে ল্যাব এক্সামের পরে হবে।
জি
ক্লাস
ম্যাম
টাইমে
এটাই বলছি
কিভাবে নিব
11টা50।
বলো? আমরা ক্লাস টাইমে তো নিতে পারবো না। আজকে শুধুমাত্র একটা লেকচার শেষ করলাম, তারপরে নেক্সট ক্লাস একটা তোমাকে লেকচার কমপ্লিট করতে হবে। উইদাউট
9
কমপ্লিটিং
তারিখেই
লেকচার
বলছি ম্যাম। হচ্ছে 9 তারিখে 11টা50 থেকে নেওয়া যায় না এটা বলছি।
I already said that I did not confirm the date time. Maybe that will be 10:10 or 11:50. I already said that. So if you prefer
জি মিস
11:50, I will like book the slot at 11:50.
জি মিস তাহলে আমাদের
ম্যাম
জন্য ভালো হয়
11:50
11টা
বেটার, কারণ
পর্যন্ত
হচ্ছে
ক্লাস
অনেকের
আছে।
পরীক্ষা আছে।
Yes, ma'am.
Okay, then 11:50. Okay, so any other
ম্যাম
question?
ওই 9 তারিখের কুইজটা ম্যাম কোন কোন লেকচারের উপর হইতে পারে? 15 আর 16।
Number 9, today's lecture and the next lecture.
ওকে ম্যাম।
Okay, today's lecture and the next lecture we'll have quiz on 9th, and that is maybe the last quiz. And we have 4 classes remaining, and whatever the lecture we will be able to complete, up to that we will have our final exam. Okay, so now I want to like have a pop quiz. So please, you can have 5 minutes to revise the last lecture, that is lecture 14. Revise the last lecture, and then I am going to give you the link. Can you hear me?
Yes, miss.
Yes, ma'am.
Okay, so revise the lecture 14, that is the previous lecture.